Welcome back. Today, we will solve together variant 42 May June 24. Okay, let's start. Fungal cells and plant cells contain mitochondria. See the function of mitochondria. As we know, mitochondria is certain organelle found in all type of cells except prokaryotes and also red blood cells. Okay. This is the structure of mitochondria where glucose and oxygen going inside mitochondria and react together to form waste gas like CO2 and H2O with the release of energy in the form of ATP. So mitochondria is involved in aerobic respiration. Okay. Please don't write respiration only, aerobic respiration. Why? Because oxygen is involved in this reaction. According to balanced chemical equation for aerobic respiration, glucose plus 6O2 reacts to give carbon dioxide plus water plus energy in the form of ATP. It's okay. State one feature of plants that is used to distinguish them from fungi. Okay, plant cells distinguish them from fungi. Look firstly at plant cell and fungal cell like yeast. As you know, yeast is unicellular fungi. What are the differences between these two type of cells? Look, plant cell contain chloroplast, while fungal cell without chloroplast. This is the reason why fungal cells are saprophytic, not autotrophs. They cannot unable to make their own food, while plant cell meat. Okay. The first feature, presence of chloroplast in plant cells. Second feature, both possess cell wall. As you know, all kingdoms, all cells for all kingdoms have cell wall, except animal kingdom and the animal cells without cell wall. Okay, cell wall of fungi made of cellulose, while cell wall of plant is made of cellulose, while cell wall of fungi made of chitin or chitin. Okay. Now, yeast is a fungus that can respire to produce ethanol. See the balanced chemical equation for this type of respiration in yeast. Yeast is fungus that can respire to produce ethanol. State the balanced chemical equation for this type of respiration in yeast. As oxygen is available for yeast, they respire aerobically to produce a lot of ATP, while at lack of oxygen, they respire with another type of respiration called anaerobic to produce little amount of ATP. Okay, so now this type of respiration to produce ethanol and aerobic respiration. So anaerobic respiration, the yeast only use glucose without oxygen, without presence of oxygen, and glucose reacts or decompose into ethanol, still containing a lot of energy in bonds, in chemical bonds between carbon, hydrogen, and oxygen. Okay, still contain a bulk of energy not released during this type of respiration, plus carbon dioxide, okay, as waste, and with release of energy needed for yeast cell activity. It's okay. This anaerobic respiration. Also, this type of respiration called alcoholic respiration or alcoholic fermentation due to production of ethanol. Yes. Now, the scientists investigate the effect of sugar on respiration in yeast cells. One flask contains 100 cubic centimeter of sugar. 100 cubic centimeter of sugar is this flask. conical flask, and another flask contains 100 cubic centimeter of water, only water. Here sugar, here water. Okay? Both flasks contain the same mass of yeast, or sure, the same type of yeast. Same mass and same type of yeast. The temperature was maintained at 25 by using water bath. Okay? Scientists use the apparatus shown in figure 1.1. Here, conical flask inside water bath at 25 Celsius, yeast in sugar solution, or yeast and water. Okay, so this experiment done twice. One flask containing sugar solution with yeast, and another flask containing yeast dissolved in water. It's okay. Now, look at this graph. Figure 1.1 is graph of the results of the investigation. Here, great significance difference between carbon dioxide gas released or produced when yeast found in sugar solution, while little amount of carbon dioxide gas released when yeast is found in water only. What's the difference? Why in presence of glucose the carbon dioxide yield is more? Oh, sure. Yeast need a source of energy. Okay, respiration means what? Release of energy from foodstuffs like glucose. Glucose is monosaccharide. Glucose is monomer of carbohydrate, is immediate source of energy. So the yeast use glucose as source of energy, immediate source of energy. Okay, so it produces a lot of CO2 in this flask. It's okay. Now, sorry. Now the same figure again. What the question? Single eye using the gradient, the graph shown in figure, calculate the rate of carbon dioxide produced by the yeast in sugar solution between 10 minutes and 15 minutes. Rate. What is meant by rate? Rate means difference over time, per time. Difference. Difference of what? Difference of carbon dioxide gas volume, okay, produced between 10 minutes and 15 minutes. Where 10 minutes here, 10 minutes, 15 minutes here. It's okay? Clock, please. Now, at 10 minutes, the volume of carbon dioxide gas produced is 11 centimeter cubed, while at 15, after 15 minutes, the yield of carbon dioxide was 19 centimeter cubed. So the difference now 19 minus 11. Over time, what's the unit now? Cubed centimeter or centimeter cubed over minutes, per minute. This for 1 mark. What's the workout? 19 minus 11 over 5 minutes time lapse, okay? Difference in time, 15 minus 10 equal 5, equal 1.6 centimeter cube per minute. It's okay? Okay. Now, suggest the reason for the oil layer in the apparatus shown in figure 1.1. The examiner or the student put a layer of oil over yeast in both flasks. Okay? Why? To prevent air entering the solution or suspension, to oblige the yeast to doing anaerobic respiration. Okay? So to allow the yeast to respire anaerobically away from oxygen, away from air. This layer acts as barrier, okay? Prevent mixing of flask's air here to enter inside yeast suspension. It's okay? Now, state one reason why no more carbon dioxide gas was produced after 35 minutes by the yeast in sugar solution shown in figure 1.1. Here's a graph. And Where is 35 minutes? 35 minutes, I mean here, okay? Log, please. Perpendicular line by ruler. Okay, after 35, so this area is flat, plateau. Why? No more carbon dioxide was produced after 35 minutes. For sure, as glucose present, the yeast cells respire anaerobically, okay, in absence of oxygen. As glucose run out, used up, there is no respiration, okay? Where is the substrate? The enzyme of yeast is ready. What about substrate? Substrate is glucose. Glucose is absent now, run out, okay? So the answer, where all glucose is used up or respired, okay, after 35 minutes of the experiment. The scientists repeated the investigation using yeast and the sugar solution at temperature of 95 Celsius, 95, near boiling. Near boiling means what? Means the yeast offshore died. Okay, explain why no carbon dioxide was produced at temperature of 95. Yeast cells died due to too high temperature. Too high temperature. Look, what happens to enzymes? Where enzymes, active sites, has been denatured. What is meant by denatured? Look, as this substrate, this active site, ABC active site depression or groove, is fat, complementary fat, at 95 or B1 optimum temperature of yeast to respire, to use glucose. What would happen to ABC active site? It becomes flat. It became flat, okay? So no longer the active site bind fat, complementary fat with the substrate, okay? The enzyme is present, but deformed, denatured. So it is unable to bind with its substrate, I mean glucose, okay? So the active site is not complementary fat to substrate, I mean glucose. It's okay? Now, state one way in which humans use the carbon dioxide gas produced by yeast cells. Okay? One usage of carbon dioxide gas produced by yeast cells. Look at this video. Both in furnace is flatted with gas carbon dioxide to carbon dioxide gas is take for rising dough, and make the texture of bread or cake spongy. Why? Due to carbon dioxide gas, due to heat inside furnace, expands, okay, and they make spongy texture for the cake or the dough. It's okay in baking. Now, state the name of one gas other than carbon dioxide that contributes to the enhanced greenhouse effect. Okay? Here, natural greenhouse effect on the left. This natural. Okay? The thickness of greenhouse gases here is little. Is very little, very thin. But it is needed to give warmth to the Earth's surface throughout the history. Okay? Along the history. What about human enhanced greenhouse effect? This is due to human activities like industries, like using certain types of fuel like fossil fuels. What happens to this greenhouse gases layer? It increased. Which criminals of this problem? Of sure, carbon dioxide here, carbon dioxide here. This the reasons why human enhanced greenhouse effect due to increased production of CO2, okay? Increased release of CO2, deforestation, okay? So what other gases contribute to the enhanced greenhouse effect? Methane gas and the nitrous oxide. Nitrous oxide, any one of them. You can answer CH4, methane gas, or nitrous oxide. Both are criminals in this problem, okay? Now, yes. Uh, Question number 2 defines the term sense organ. Sense organ. Sense organ like this figure, 5 senses, eye for sight, ears for hearing, touch, skin for touching or touch, nose for smell, tongue for taste. What is meant by this sense organs? Why they are called sense organs? Because they contain group of receptor cells, ending of sensory nerves, okay? The group of receptor cells, that what? That detect and respond to certain stimuli. Detect change in external or internal stimuli, like sensation of hotness, coldness, pain, touch, pressure, all of this, or chemicals like nose, and so on. Group of receptor cells that detect and respond to certain stimuli. Letter B, figure 2.1 shows the structure of the eye. It also shows the pathway taken by the nerve impulses, which help bring about changes in the eye in order to bring light to focus. It is big chance to revise all items of anatomy, eye anatomy or structure of the eye. Letter A, huh? Yes, retina. Letter B, this depression is called fovea. Good. Letter C, at the beginning of optic nerve, it's called blind spot. Okay? That's where fibers pathways. Here, optic nerve. Please don't write nerve. Optic nerve. Okay? Here, associated with lens, okay, from above and below, like this dot. This is called ciliary muscle. Ciliary muscle, okay? Then lens, then in front of the lens, eye pupil opening, allow light to pass through. Cornea, transparent tissue without blood vessels, okay? J, the pretty part of our eyes, colored part of the eye, iris, yes. K, suspensory ligaments above and below the lens, okay? Above and below the lens, suspensory ligaments, okay? Now the question. Draw a label line and a letter X on figure 2.1 to identify a motor neuron. As you know, a motor neuron bring nerve impulses from brain. Bring nerve impulses from brain to what? To effector, like muscle or gland. So brain here, bring impulses from brain. So here and here, look also here, here, to reach what? To reach ciliary muscle attached to the suspensory ligament and also to the lens. Look. Now X here. This identify a motor neuron. This motor branch of motor neuron. It's okay. X here. Now describe how the events that occur at a synapse generate an impulse in the next neuron. Synapse. Synapse is a gap between two neurons or neuron and the muscle. Two neurons, successive neurons, okay, or neuron and the muscle, okay? This synapse. How events that occur at synapse generate an impulse in the next neuron. transmitted to the next neuron. How come? Look, please. Look at this short video. Okay. Okay. Now, with discussion. The cell above is called the presynaptic neuron. Presynaptic neuron. Okay, this neuron, terminal part of previous neuron. Below, second neuron. The area between them, okay, called synapse or synaptic gap or synaptic cleft. Synapse, okay, for sure, synapse. Now, look, the presynaptic neuron contains vesicles, spheres or spheres, yes, filled with neurotransmitter. This spherical molecules green in color, neurotransmitter. What happens when nerve impulse reach the area of this swelling of presynaptic neuron? It triggers the vesicles containing neurotransmitter to move down towards the presynaptic membrane. Okay? Look, please. Here, presynaptic neuron, synaptic vesicle containing neurotransmitter. Okay? Now, these vesicles reach the presynaptic and diffuse and release neurotransmitter in the gap. This neurotransmitter is specific molecules. And reach what? Reached receptors, protein receptors specific to bind with this neurotransmitter at the second membrane, at the membrane of both synaptic neuron. Both synaptic neuron. It's okay? This receptors, these green areas inside the membrane, involved in the membrane structure of both synaptic, is called receptors. Look, these receptors. This receptor, this protein receptor has a gap, has a channel. When the receptor receives neurotransmitter, this channel open and the nerve impulse passes to the postsynaptic neuron. Okay? Okay. All of this vocab or key terms must involve it in your answer to bring full mark. Okay? Presynaptic neuron, postsynaptic neuron, presynaptic neuron containing vesicles containing neurotransmitters hormone. Okay? Both synaptic contain receptors to receive these neurotransmitters and open to trigger nerve impulse in the second neuron or next neuron. It's okay? Now describe and explain the process of accommodation. Accommodation in the eye to view near object. One of the functions of the eye is accommodation process. Accommodation means focus for near object or far object, near or far. What happens if we put this marker in front of our eyes? The light ray is coming from near object, need more refraction, okay, to pass through our eyes to focus on fovea for sharp vision. It's called accommodation. How this bring about? How? Look, please. Accommodation, focusing, double C, double M. Accommodation, double C, double M. Accommodation. Okay? Look, please, near object. Close focus. This our eye. The light coming from near object, near our lens, near our eyes. Okay? Sharp in angle. Okay? How this light rays, instead of pathing up and down like this, focus on fovea here for sharp vision of a near object. How come? It need more refraction. Look, please. Ciliary muscle contract. Where is ciliary muscle? Here. Ciliary muscle in this part. In this part. contracts. Okay. Then suspensory ligament must slacken. Suspensory ligament, okay, above and below the lens, attached to the lens directly, slacken, loosen. Okay, so it allows the lens to become fatter, to become horizontally thicker. Look, please. Suspensory ligament slackened. Lens is not pulled, not tight, slacken, loose. Okay, slacken ligaments. So lens is not pulled, allow the lens more convex. Instead, slight convex, more fatter, to allow more refraction of light rays. This lens is fat. Now, light rays are bent strongly. Light rays focus on retina or fovea of retina. It's okay. This accommodation when our eyes accommodate with view of near objects. Okay. The reverse of far object, light rays coming parallel. So no need for more refraction. Less refraction is needed. So the lens become thinner, become flat. How come? The suspensory ligament become tight, stay tight, okay? And ciliary muscle relax, so the lens become less thin and less refraction, and light rays passes parallel and less refraction needed for this light rays, and they met at fovea for sharp vision also. Okay. Now describe the distribution and function of rods and cones in the eye. Look, please. He asked about distribution and function of rods and cones. What about distribution? First, distribution. Here, this graph shows visual angle degrees from fovea, away from fovea, 0 angle at fovea. So this part express fovea, where at fovea cones are accumulate with high concentration of high number of cones. Look, maximum number of cones found at area of fovea. No rods at fovea, but all light receptors or photoreceptors are cones at fovea. No rods. What about rods? Rods are found, okay, away from fovea with decreasing number, okay, from high here and high here, and away from fovea, decline in number and decline in number. So cones are accumulated throughout the retina. Rods are found or distributed throughout the retina, while cones are accumulated at the fovea. Rods and cones not found at all at blind spot. Here, this empty area means no rods, no cones at blind spot. Again, with this table. Now, here for distribution, firstly, for rods. Distribution of rods found throughout the retina, yes, but none in the center of the fovea or blind spot. Okay. What about cones? Cones concentrated in the fovea. Concentrated. While little cones away from fovea. Throughout the retina, but with little number. Okay. Like here, this bluish area. Little number of cones found in the retina. Accumulate with high concentration at fovea. What about the function now? Rods sensitive to low light intensity at dim light, okay? At sunset, we can depend on rods. For what? Detect white and black figures, okay? Comments. Provide us with night vision when we can recognize shapes but not colors. This roles, function of roles. What about cones? Sensitive only to high light intensity at daytime, okay? Or intense light, intense E light. Detect what? Detect the color. Do not operate in poor light. In poor light, I mean darkness, okay? Comments. There are three types of cones. Three types of cones. Green, red, green, and blue. Okay, found in the written. It's okay. Now, red-green color blindness is sex-linked characteristic. It is controlled by a gene on the X chromosome. Sex-linked characteristics, okay, linked with sex, linked with gender of person. So their genes may found on X or Y chromosome. Here, according to our syllabus, all cases for sex-linked traits or characteristic is controlled by gene on the X chromosome. Color blindness, red-green. Color blindness is sex-linked characteristic. It is controlled by gene on the X chromosome. There are two alternative versions or forms of the gene, of this gene. Sorry. No color blindness, X A capital, color blindness, X A small. X is a chromosome. A is the allele, A capital or A small, alleles of the same gene. C is the term used to describe an alternative version of gene. And they are called alleles. Okay, allele. Allele, like blood group, IA, IB, IO. Three alleles for I. I is the gene. A, B, O alleles. Three different alleles for one gene, the gene for blood group in human, for example. It's okay? Here exactly, but these genes, these alleles are found on X chromosome. X chromosome. I mean X chromosome, this sex-linked characteristic. Again, now see the genotype of a male with color blindness. A male with color blindness. What is meant? What's the structure of male? XY. Yes, XY. What about color blindness? Color blindness means he has a small over X carried by X like this. This is the genotype of male with color blindness. X is small y. X is small y. From where this X is small gene or allele coming from? From father or mother? Of sure, from mother. So this affected son, okay, inherit this defected allele, I mean X is small, for color blindness character from his mother, while his father gave him Y chromosome. Y chromosome doesn't carry any genes responsible for this character of disease. It's okay? Now with question 3. Meiosis and mitosis are important processes in the life cycles of organisms. Figure 3.1 shows the life cycles of two different organisms. Organism 1 has a simple life cycle. Organism 2 has complex life cycle. It has stage A that produces spores and stage B that produces gametes. In the diagram, the haploid number of chromosomes is represented by N, 1N. The diploid number of chromosomes is represented by 2N, diploid. Okay? Organism 1. Organism 1 here, adult with 2N. Process B to produce egg and sperm in two different individuals. Here, process W, forming zygote, develop into embryo, then by process Q, develop into adult again. It's okay. This simple life cycle of organism. For organism 2, here multicellular stage A. Multicellular stage A, diploid number to N. By process R, it forms spores 1N from 2N to 1N. Then spores change into multicellular stage B with N also, but multicellular. As you know, spores unicellular cell. Okay. Now multicellular stage B, by process S, it forms female gamete N and the male gamete N also. Okay. Fuse together to form zygote. Then T, by process T, the zygote change it into embryo, then multicellular stage A. It's okay. Now, table 3.1 shows the letters B to Q, B to T, sorry, B, Q, R, S, and T. In figure 3.1 and type of nuclear division, place a tick right in each row to indicate the type of nuclear division that occurs at each of the letters B to T. Here. Look, please. Now, this organism 1. What about process B? Here, process B to form egg and sperm. So, this meiosis or mitosis 2N change it into sperm or egg. As you know, egg and sperm are gametes. This gametes in this individual haploid. Haploid. So 1N, 1N. So the process B is what? Process B is meiosis. It's okay? Meiosis to form gametes. What about Q? Where is Q? Q here. In the same organism 1. What about Q? Q before, okay. Q, embryo, develop into adult. By which process? Like human, for example, by mitosis, millions times or runs of mitosis. Okay? Mitosis. What about then R? Where is R? R here. R, we start with 2N, multicellular stage, to form N spores. The number of chromosomes reduced into half. So this process R is considered meiosis. Yes, right, meiosis. Having the number of chromosomes. What about S? S is new idea for you. Okay, you know that gametes formed by meiosis. Here, exception. Look, please. Multicellular stage B, N. Form gamete, NN. So N keep its number of chromosomes. Although they keeping their number of chromosomes, this process as form what? Form female and male gametes. So these gametes here in this case are formed by mitosis. By mitosis, not meiosis. It's okay. This is exception in this individual of organism. Okay. Now 14. Now 14. Zygote develop into embryo of sure like human, for example. Zygote to N form embryo, still to N, form multicellular stage to N. So this depend on mitosis. Depend on mitosis. It's okay? Okay. Now, see the name of process. Just I need to mention something important in this figure. In organism 2, like ferns, life cycle, they reproduce by spore in certain part of its life cycle. Okay, like ferns. Ferns, this advanced plants, they reproduce by forming spores in certain stage of its life cycle. Okay, in chapter 1 in classification, we mentioned that. Now see the name of process W, shown in figure 3.1. Where is W? Here, W. What's the name of process W? Where egg nucleus fuse with sperm nucleus. This is called, yes, fertilization. Fusion of egg nucleus with sperm nucleus. This is called fertilization. Now, an embryo contains stem cells. An embryo contains stem cells. Complete the sentences about stem cells and body cells. Stem cells of embryo are something called unspecialized. Stem cells in embryo, after zygote, after few days of zygote formation, implantation of zygote and cells coming from zygote by mitosis, implanted in uterus. Okay, these cells contain stem cells. This embryo contains stem cells. This cell is able to give all body organs, all body cells, all types, so they are called unspecialized. Unspecialized or undifferentiated bulk of cells, but undifferentiated, no difference. Okay, all the cells are the same, but unspecialized, undifferentiated. Okay, we can mention either one, undifferentiated or unspecialized. That divide by what? Embryo cells divide to form fetus by mitosis. To produce daughter cells that can become what? Daughter cells later on become eyes, become nervous system, become skin, tongue, and so on, muscles, and so on. And to become what? To produce daughter cells that can become specialized, differentiated, different. Okay? Now, specialized or differentiated for doing certain job, certain function, specific function. Like what? Here, single cell, like cells of embryo, undifferentiated. May its fate, its future. Stem cells able to replicate itself. Okay? Also, during replication, some of this resulting cells become stem cells again, and the others become differentiated into specific cells, like what? Like a neuron here, like epithelial tissue, like red blood cells, like fatty cells, like white blood cells, like muscle cells, and so on, differentiated into many cell types. Okay? This is the definition of stem cells. Stem cells are unspecialized cells that divide by mitosis to produce daughter cells that can become specialized for doing certain job. Most body cells in an organism contain the same genes. Same genes. We have in each cell nucleus about 30,000 genes. Okay, 30,000 genes. Okay, but many genes in particular cell are not what? Many of them are inactive, are switched off. Okay, so they are not what? Are not expressed. Are not expressed. Are not what? Switched on. Okay, because the cell only makes the specific protein it needs according to the speciality or the differentiation of the cell. Say, certain cells in pancreas produce insulin. The gene for insulin in this pancreatic cell is switched on. Okay? The same gene is found in skin, but not produce insulin. Why? Because it's switched off, not expressed. Okay? It's okay? Now like this. Here, around 30,000 genes are present in each nucleus in each living cell. Okay, human. Here, gene A switched on. Switched on means what? The gene here, area or section of DNA, is switched on, means it's transcribed mRNA. mRNA released from nucleus goes to cytoplasm, okay, and translated by ribosomes to produce certain protein, certain polypeptide. Okay, this gene expressed like insulin after having a meal, it's expressed, but switched off during fasting. During fasting, the same gene, insulin gene, is switched off, not expressed. Okay, here gene B in the same cell may be neighbor to gene A, but is off now. It is not needed in this cell at this time. Maybe needed later on, maybe. May the cell is not specialized to produce certain type of protein if the gene B is expressed. Not needed, not specialized. Okay? This make differentiation of cells and tissues in our body. Now see the events in the life cycle diagram for organism 1 in figure 3.1 that would not be present in the life cycle diagram for asexual reproduction. This comparison between life cycle of diagram of sexual reproduction and asexual. Asexual reproduction has no events from this diagram. Like what? Like asexual reproduction is characterized by what? By no what? No process B. No gamete formation. Okay? No haploid cells are produced. No gametes, no egg, no sperm. Okay? No formation of gametes, egg or sperm. No fertilization. No zygote is formed. No embryo is formed. It's okay? No embryo is formed. We finished 3 questions out of 6 questions in this exam. Thank you for watching. Meeting you next time soon, inshallah. Good luck. Bye-bye. Have a nice day.